get approval. So when we talk about the nanotechnology, there is no well accepted or standard definition of nanotechnology. For example, if there is a particle or feature which is 200 nanometer of size, so whether that would be considered an nanotechnology or nanobiotechnology. So that's there is no standard opinion about that. So some people think, most people actually think that anything which is from 100 to 1 to 100 nanometer, that is under the umbrella of nanotechnology. But some people don't agree with that opinion. So in general, anything less than 100 nanometer can be considered in the nanotechnology range. But if there is something like 110, 100, 150 nanometer, so I would still say, according to my opinion, that was still in nanometer range. So, but definitely if there is something 500 nanometer, so in other words, a 0.5 micron, maybe we should not call that a nanotechnology, okay? So that's normally how people define anything from 1 to 100 nanometer is a nanotechnology. There are different techniques which can be used to make structures and features in that range, and which those structures and features and devices can be used for a lot of different applications. As a one example, we can use optical lithography, which we'll cover later on. What is optical lithography? But basically, it's a technique which you can use to print devices or print features on the surface. It can be your silicon wafer, it can be a glass surface, or any surface which is flat. You can use this technology, optical lithography, to print features. And by the end of 20th century, actually, people have achieved up to 100 nanometer feature size with this technology. And right now, people who are making silicon IC fabrication devices like transistors, I know the Intel and IBM, they are working on technology node which is about 7 nanometer, and one is working on 5 nanometer. So it means they have a capability to make or print features in the range of 5 to 10 nanometers. So that makes things very, very small, and it means we can have even more processing power, even more number of transistors in a chip, and then it can be used for making computer devices, cell phones, and all that, where we have CMOS devices nowadays. Just give you some example. When we say size, what are the different structures and molecules which are available in different sizes? So let's say if we look at the retina implant, their size is normally in millimeter, 10⁻⁻3. There are also MEMS, some MEMS devices. So MEM is a mechanical electromechanical systems. So these are the devices you might have heard about, BioMEMS or MEMS. So those devices are actually, all of them, they are at a micron scale, and you can make different structures with the MEMS. And they have, again, this is a technology which can be used for a lot of different applications. So anything which is on a micro scale and you have some electrical and mechanical components, that's called MEMS. And this is one example where you have a gears and they're doing something with that. Just to show you, this is SEM image, scanning electron microscope image. And then our human hair, as we discussed, is about 40 to 50 or 60 micrometer. It's also in a micrometer range. Bacteria and a lot of other fungi, cells are also in a micron scale range. Our human blood is also in micron scale. For example, bacteria is about 3 to 5 micron. E. coli, for example, is 3 micron in length, and the width is about 1 to 2 microns, so because it's in a cylindrical form. But yeah, most of the pathogens, bacterial pathogens, are in this range. And cells are also in this range, but although there is a high variability between cell sizes, For human cells, the smallest cell is about 4 to 6 micron, which is red blood cell. Like 1 dimension of a red blood cell would be the smallest one. And the largest human cell can go to actually even 50 micrometer in the case of cancer cell, which is pretty big. But that's rare. Most of the cells are in 20, 30 micrometer range or smaller, or 10 micrometer. Most of the white blood cells are 10 to 12 micrometer, but they are ranged from 8 to even 20 micrometer. Okay? Some of them are pretty large. But most of the human cells are in micron scale range. And then there are some man-made and also some natural species which are structures which are in nanometer. For example, this is a buckyball, this is a carbon nanotube structure, and they are, because it's a nanotube, so it's actually on a nanoscale. And then the viruses, as we discussed, most of the viruses are in a nanometer range, but some are actually larger than that, as the one which we discussed last time was HIV, which has an average size of about 100 nanometer. So that is not in this range, but close to that range. So, but most of the viruses should be in 3 to 15 nanometer, but some are larger also. So do you remember how much was a size of Zika virus? We discussed in the previous class lecture. You forgot? For Zika virus, so let me give you option 30, 50, 70. 30, 50, anyone for 70 nanometer?
Why not?
Yeah, it was actually 50, okay? So 50 nanometer is the size of approximately 50 nanometer for a Zika virus. And for human DNA, I told you the length of a DNA is very long, but the diameter of a human DNA is about 1 to 2 nanometer in terms of size. If you take a DNA and actually put it in your hand from both sides, the diameter of the double helix, which you can see here, ATGCs, it is about 1 to 2 nanometer. So yeah, we have stuff which is actually on nanometer scale, and there are hydrogen atoms present in a molecular form. They are also in a nanometer range. So atoms are definitely, most of them are in nanometer. A couple of other things which are present in a natural environment, and there are also certain things which we have made actually on a micron scale with the technology. So some like DNA, I don't know, it's not clear. What is this? Can you read it? What is this? What? Okay, atoms of silicon spacing. So yeah, that's also present naturally. Maybe that's why it's put there. It's a TM image, looks like. So it's a transmission electron microscope image, and you can see how much resolution we have with that microscopy. Even you can see the atoms very clearly with that imaging technique, but that's definitely very expensive. On the right side, there are certain stuff which actually humans have made with using a technology. So this is a very famous image from, I think, IBM engineers. They use a scanning tunneling microscope tip to arrange atoms on a surface. And in another image, actually, they have written IBM with that. With the atoms, you can write, you can move atoms one by one. So that's the capability which we have so far with the technology. But the problem is, like, those processes are very slow. When you really handle atoms with the tip, you cannot do that process for with a lot of efficiency. So it takes a long time to do that. Even, let's say, for example, in imaging this kind of a structure, it takes about 1 or 2 hours. Okay, so just taking a couple of images, it takes a much longer time to prepare your sample accordingly and then image it. And images, so this is a very nice image, by the way. So carbon nanotubes definitely are what our technology has given us the power to make it. But on the other hand, the carbon nanotubes are now started. People have started using carbon nanotubes in different applications, and one of the application I remember, as also shown here, is nanotube transistors. So if you're familiar with the transistors, then you will understand it. If not, then maybe you will not grasp the basic idea where we can use the carbon nanotubes. But carbon nanotubes are used to make a channel region of, so the MOSFET or the transistor has three different regions. Source, one is source, which from where we get the electrons. There is a drain. The electrons flow from source to drain, and there is a channel region which connects source and drain. So the silicon carbon nanotube is being used as a not commercial products, but is still in R&D. They're using carbon nanotubes to make FETs or CMOS devices, and they're also actually MEMS. And just to give you an idea that the head of a small pin is about in a millimeter range, 1 to 2 millimeter. It depends on the size of pin. Okay, so maybe last slide on showing you some more structures which are in nano and micrometer scale. So for example, if we look at these structures, let me show you the whole slide. So if we look at these structures, we have some organs, tissues, and some species also in a millimeter range, and then it's a human blood, plant and animal cells are in that range. Bacteria is also in micrometer, and then we have viruses, we have proteins, DNA, and different bonds between a carbon or a hydrogen, so they are in nanometer or sub-nanometer range. On the other hand, we also have technologies which we have made by using different methods, which have quite a few applications. For example, specifically talking about structures which we have designed by using technology, which are nanometer. One example is a 35 nanometer MOSFET. It's a fast of MOS-T. So it's actually MOSFET means it's a transistor. The image is, there are a couple of different structures. So this is the gate region, this is source, this is drain, and channel is at the bottom. So, but the idea is, yes, we can make structures on nanometer, and we can make it with a good repeatability and efficiency, and that's why we can print billions of transistors with a good efficiency. And once we make a chip, it still works for years. Let's say your computers, we have all of us have transistors, billions of transistors, so they are very reliable, and they work for a long period of time. Then also there are some structures called the nanopores, for example. So nanopores is, as clear from the name, it's a pores of a size of nanometer. So nanopores have been actually developed with actually one application when it started is for DNA sequencing. So the idea was that to make a pore of a nanometer size. So how much was the size of thickness of a human DNA? 1 to 2 nanometer. So think about it. If you have a pore which is a 1 micrometer scale, and if let's say we make a chemical cell where you have 2 electrodes and everything is filled in water. So you have 1 electrode on this side, and the other side you apply some voltage, and if it is electrolyte, there will be some current flow, right, through this pore, because there are 2 chambers which can only connect through this pore which we have. So if you apply, let's say, 1 volt, so based on the conductivity of a solution and the size of a pore, some of the ions will move from towards anode or cathode. So what happens if the pore is now smaller? So will you see a change in conductivity or change in your current? Yes, because it will not allow enough charges or ions to move through it, right? So now think about if you have a micron scale pore, only 1 pore, so there is no other way the charges can travel from one chamber to the other chamber. If you have now a micron scale pore, and if there is a DNA, which charge DNA has? We talk about it. DNA is positive and negative. So DNA is negatively charged. So in that case, if we put a DNA to one side, which is a negative electrode, and apply voltage, so DNA will try to go towards the positive side, right? Based off under the potential. So when it goes to the positive side, it has to pass through the pore. So once it's passed through the pore, if it is a micron scale, will you see any fluctuation in your current which you are getting? No. Because the pore size is very big and the charge which is transferring by a nanopore is very, very small. Because it's very, very small, it will not physically block the pore. Let's say now if your pore is 10 micrometer and if there is a cell 8 micrometer. Now if cells passes through it, it will actually physically block about 80% of the pore, right? And then you should be seeing less current during that time. So actually if this is your current, let's say 1 ampere, and when the cell will pass through it, the pore will be physically blocked for a very fraction of time. So you will see a dip in current, and then once it's done, it's on the other side, then again the current will come back to the normal state. So in that way, you will get actually pulses as the current is passing through it. But now if there's a DNA which you want to detect, you have to make things on same scale. So you have to pore which is about 5 nanometer or around 10 nanometer, even smaller than that. So then if there is a 5 nanometer pore and if 2 nanometer thing is passing through, yes, you will see some dips. So that was the basic idea. So the goal was to actually, the goal was actually to sequence the DNA like that. So because DNA has 4 bases, A, T, and G, C, and the goal was when DNA is passing through it, maybe each base will give us some different signal, and in that way we may be able to decode the sequence of a DNA. So that would be a very straightforward way to do it. So far, there is a limited success in that area. You can look at the company. Now they are selling some nanopore-based devices for DNA sequencing, but it's not very straightforward how they do it. But it's still they use nanopores for those sequencing applications. But there are a lot of limitations also of this technology, which we'll discuss later on when we cover this topic in a little bit more detail. But for now, you can look at the Oxford nanopores. Oxford is a UK-based company, and now they are doing very well in the business. And most of the technology is based on the nanopores. And our nanopore-based devices are commercially available very recently, and I think a year back or so. So it's a very, very new technology. So, but the idea is, yes, you can make, to make a pore in a film, which is only one pore, 5 nanometer, is not an easy task. It's really challenging, because to make a pore with a good reliability and make sure it's a 5 nanometer only, so it's complicated. But there are ways to do it, which we'll cover later on. So, but the whole idea is that, yes, we need, for various applications, we really need features and structures which are at a nanoscale, because large scale structure doesn't work. This is one example in sensing. Yes, and there are quite a few examples also in other areas like therapeutics and even in nanophotonics. So we briefly talk about lecture and talk which Richard Feynman gave in 1959, I believe, and there is an article which I already have uploaded, okay, on a canvas. So you might have read the article, maybe not. So, but there is a homework coming up for related with that article. So you should read the article, and because in the article he he predicted a couple of things, like how we can manipulate nano and micro scale things, species on a small scale, and then he also talked about writing on a pinhead and a couple of other things. So your homework is actually due next week, so I'm giving you a week for that. So make sure that you read article. So it's just a reading article, and write it just one page, not more than one page at max. So one page. So, and what you have to include in the homework, the question is how much of his vision has come true so far. So when you read the article, you will understand, because you are from a different disciplines and departments, but you will you can easily correlate some of the things which he's talking based on your background with your department or your expert area. So you may be not able to comment on something which is in biology or engineering, but you should be able to comment on some of this stuff. So based on your own knowledge, you don't really have to do a lot of research in this homework, but only you have to read an article and write it, how much of his vision has come true. And you also have to comment while answering that question. So what are the things which has not been demonstrated? And let's say if he has talked about certain thing that we can have maybe nanoscale robots which are moving inside a human body and they can do different stuff. For example, if there is a discussion about that in his talk, but that I have not seen that yet. Like robots going inside a body by themselves and doing some stuff. Okay, so like on nano robots, there are some nano robots people have made, but they don't do much. So people have shown that they can make a nano kind of robots which can move slightly from one place to other, but there is nothing out there which people are using with that kind of nanorobotic technology. Okay, but if you think, that's what my knowledge says, if you think that is not correct, for example, that there are certain stuff which people have done, and you have to definitely give a reference. So in this case, you can give some website reference, which should be fine, because we will be talking about very new technologies. So basically, you have to comment on how much of his vision has come true, what are the things which he has talked about, and now you see that in your daily life or in your research, and what are the stuff which actually have not reached the level which he's talked about, and whether it is possible or not possible. Because he might have talked about things which, according to one person, may not be possible. And it may be possible based on your reasoning. Why you think it's not possible? Okay, just write a comment based on. And if you submit it, you will get maximum score. So let me put it that way. So don't get afraid that, okay, you cannot answer that question very clearly, or maybe you don't have a background. So just write it down, whatever you think about it. Okay, you can do a little bit of research if you're confused about whether this has been done or not, but in general, just read the article, lay down what you think has been achieved and what has not been achieved. Okay, so just a one page, it can be even less than a one page, just 2 paragraphs, things which have been come true, and things which which you think may not work according to your understanding. So you should bring that in a hard copy for all the people who are taking this class for in-class sessions, for people who are taking this class for as a distance learning students, so they can submit it through actually Canvas. Yeah, so they can submit through Canvas, because it will be more easier for them to do that, instead of for those people who are taking this class for as a distance learning option. Okay, and there are quite a few students, I think 8, 9 students. So, but who are registered for in-class sessions, they have to bring a hard copy. Okay, in class.
So do we follow like in like Chicago or APA or
No, just you can choose any format. Like font, for example, you can use 12 times the Roman Calibri or any other Arial, that is fine. And so there is no specific guidelines about the formatting. Yeah, for font size 12, 13, 11 is fine, but don't use 20, right? So in order to fill up the whole page. So
single or double space.
I'll leave that to you. If you think that you can convey what you want to say in less sentences, then you can use double. So yeah, so filling up the paper is not a point here. So I want you to actually read it and write it about it. So if you think that you can do still a single space, and you can maybe fill up half of a page if you conveyed your idea, but one page should be good enough. So as I told you, if you submit and if I see that you put an effort, you will get a maximum score. So that's the point. So now it's your call how you want to do it. But make sure that you do it. Just don't write, okay, I read it, I don't understand anything. So that should not be your answer. So just say something about it. And that will be a very interesting article actually to read. Okay, so Now let's move. Okay, so far we talk about just the nanotechnology and with some application in biotechnology. So when we say nanobiotechnology means we are more focusing on the application of nanotechnology in biology. That's what called the nanobiotechnology. So it's the intersection of a lot of different disciplines, as I told you in the previous class. You will see some of the concepts which will be new, and some you might be very much familiar already based on your background. Some people call it as bionano or nanobio, so it's actually a similar thing which you are referring to. So when a natural thing like a DNA, for example, or protein is used to make structures, it's called the bionanotechnology. This is what people normally accept as a definition. So when you use natural molecules like DNA, actually there are quite a few people who are making structures with the DNA. So they can manipulate DNA, they can make different 3D structures with the DNA based on how you design the DNA. And those structures, so far there are not a lot of applications for those structures, but we have a capability to make different kind of, like you can make a star with the DNA. So what's the purpose of it? No, right now we're just demonstrating that there is a technology we can control the DNA structure, we can build 3D devices, or maybe at one stage you can make maybe nanoparticles with the DNA. So you can make a structure of the DNA such that they're nanoscale, and it can have quite a few applications. But right now it's again in the research and development, so you may not find a lot of applications, but yes, people are using DNA and proteins to make structures, and when they do that, we call that as a bionanotechnology. But when we use artificial materials like silicon or gold or other things to make structures, we use chemical processes or other material science based technologies to make structures from synthetic materials. That's called nanotechnology. And if you find the application in biotechnology, we call it nanobiotechnology, but there is no hard and fast rule. Okay, but you should know that it's a bionanotechnology, so maybe we're using biological structures to make nanotechnology stuff, and if you're not using natural DNA and proteins, or you're using silicon, then you can call it nanobiotechnology, okay? But again, there is no hard and fast rule for that. So when we have intersection of engineering and biology, there are quite a few new applications which you can find and which you can easily address. For example, in this case, it shows you a couple of devices which have been used for quite a few applications. For example, in this case, it shows this is a glucose meter, right? This is a glucometer. These are, I think, paper strip-based assays, these are paper strips which you can dip it in some solution and you can detect different ion concentration. And then there are also some pregnancy test strips also available. You can find that at CVS and Walsey. Now everyone uses it. So all those technologies, you can see those products only because there are engineers and scientists and doctors working together to build those devices. And when we look at the pregnancy test, for example, if you're not familiar, so this test is actually used to find out if someone is pregnant or not. And we look at the HCG level. So HCG is actually released. It's a hormone when egg is fertilized and attached to the uterus. So basically, when oocyte or egg in a female is fertilized with the sperm, then this HCG hormone level increases a lot. And normally we test it with the urine. So if you put a few drops of urine on this strip at certain place, and if someone has a higher level than normal HCG in their urine, so that means she most probably will have a pregnancy. And these kits are normally 99.99% accurate actually, and they say it on their label, so they're very, very accurate. And then you can see a test line. And there is a test line, and there is also a sample line, a control line. So when you see the test line is also you see a color in test line, it means that you have a high HCG level, so you might be pregnant. But in other case, if you don't see the line, or if it's very dim with low contrast, so maybe then it's not clear. So maybe you might be pregnant, may not be. But the whole point is, but when you use a strip, it looks like very simple. It's very cheap. Also, not very expensive. Maybe $5 you can get one. But there's a lot of nanotechnology inside even that pregnancy strips. Do you know what is on nanoscale in that strip? If you're familiar with the technology, it's called the lateral flow assays, by the way. So there are also a couple of other assays which are available. The technology in general is called lateral flow assays, and pregnancy test is very commonly used. Anyone familiar how those devices are made? So antigen antibody conjugation, yes. So we use some antibodies to capture specific hormones and antigens. And those antibodies are definitely a nanoscale. But what gives that color? What makes that line formation? Why we see that line there? If someone has a high level or not? Sensors? No, it's optical. You just give you contrast, right? So we just... No, it's not fluorescent because fluorescent you need a special frequency of wavelength actually to excite it, right? But we see it with the naked eye, so definitely it's not fluorescent based. It is a colorimetry test because that's why you see a color change. Yeah. So in reality, they use gold nanoparticles in that test strip. So when we think about the gold nanoparticles, we think, okay, they might be very expensive. They are. But think about nanoparticle, let's say 10 nanometer. And it's a very, very, very small particle. And you can have many of them at a very low cost. So that's why these pregnancy strips, although they use gold nanoparticles, but they're very cheap. Because you're not using a lot of gold in there. So the thing is, they take gold nanoparticles, they functionalize it with some antibody, and whenever there is a more level of a hormone, so we'll talk more about it later on, but basically the antibody-based particles bind with the hormone, and then it's passing through the strip where you already have another antibody, so that also bind with the hormone, so you see aggregation of the gold particles there. When you see many of gold particles, nano-sized, at high concentrations, so they aggregate. When they aggregate, you see a contrast, and that's why you see a strip there. Okay, so yes, there is a nano. If you use particles which are micron scale, then this may not work. And you know why? Because if you use micron scale particles, and this whole strip is made on a paper. These are also called the paper-based devices. Like some devices people nowadays are using, they are made up of paper, cellulose paper, which we use for writing. Because this paper has certain properties, like it's microporous. If you see it under the microscope, you will see there are a lot of pores and fibers around it. Okay, so because it's a microporous, so if a regular paper normally is a hydrophilic, means if you put a water drop, those porous structures work as capillaries for the water. So that's why the water will go inside a paper very quickly. But if it's hydrophobic, that's a different case. Like if you treat a paper with something, so to make it hydrophobic, then it will not seep into. But in general, that property is called the wetting property of the papers. And because of the wetting property, if you put a solution, and that's the case in here, when you take a urine sample, put it there, it automatically goes to the other side. You don't really have to push it. So it's a passive flow. So you don't have to use the syringe pumps or other methods to actually push a liquid from one place to other place. And that's a very big benefit which we get when we use a cellulose paper base. We make cellulose paper based devices. So because then you can make those devices for a point of care applications like where, or maybe at home diagnostics, you can develop that with the paper. And those fibers and the paper structure, so it has micropores, which is the difference, and there are quite a few types of papers which are available. So now think about it, if your paper has a micron scale features, and if you use a microparticles, so there are good chances that micron scale particles will not be able to make it to the test line, because they will block the pores very easily. But now if you use a particles which are 10 nanometer size, so they can easily pass through the pores. And that's why those particles, from where those particles come, they are already actually deposited in certain area. When urine comes, it flows the particles with them. It makes there is an interaction between the hormone and the antibody, and then the particles flows with the liquid, with the urine. And then when there is a test line, if there is a hormone capture, so it will start aggregating. So yes, so that's why if you really want to develop these kind of devices, you really have to work at the nano scale. Means make nanoparticles, how to make it, how to make sure that they are stable and all that. So that's a different story. But yes, the nanotechnology, you will see it around you all over nowadays. There are also some glucometers and other assays which we have talked about previously, so I'll not go into detail in the previous lecture. But yes, you can you can also make these are micron scale, so you can make this is a strip which is used for glucose sensing or sugar level sensing in your blood. So you put a drop of blood here on the strip, and it will give you some reading, and that tells you how much glucose level you have with some units. And actually this strip is just electrodes. There are some electrodes already printed with a micro or fabrication technology. So you make those electrodes very small because you don't want big electrodes, and you also you want to make sure that you have control on their dimensions every time you get a similar device. And so whenever, again, there are some enzymes which are deposited which react with the glucose and change the conductivity of a solution, and then you actually are reading just the current. You apply some voltage and measure the current. That's what this device does. It's an ammeter. Means you are measuring the current by applying a voltage, and with the measuring current you can then correlate, okay, if this is a sugar level, so then this much current you are getting, so maybe most probably this is the sugar level in the blood. And these are normally very reasonable and devices or glucose meter ones, so you see some fluctuation in the results, maybe 5 to 10%, but that is pretty acceptable. Okay, these are some of the images, more images which we have made for the nanopores. Like this is from one of my article. When I was working as a PhD student, at that time we designed some methods to actually shrink the pores. I just want to show you some nice images. So in this case, we started with a pore which was about 400 nanometer or even larger than that, and then we shrink it to even a pore which is 3 nanometers small, 3 nanometer in diameter. So this is a really small pore which we made in the middle one. And so there are methods to shrink the pores and make things small in this in nanopaper-based technology development. And this is one example how we can use the carbon nanotubes to develop sensors in this case. So this component can be considered is actually a transistor. You have a source here, you have a drain, and you connect the source and drain with the carbon nanotube, single carbon nanotube, and then you functionalize it. So what happens when you apply voltage across these two terminals? So you get some current, and that current will pass is through that carbon nanotube channel. Okay, so we call that a channel, because if you don't have a carbon nanotube there, you will not see any current. So in this case, you have insulator at the bottom, and when you apply voltage, and if there is no carbon nanotube, will be a gap between those two electrodes, so you will not see any current. But when you have the carbon nanotube there, and that's how you make this structure, then you will see current flow through the carbon nanotube, and that's why we call that carbon nanotube as a channel. Now think about if we have now proteins which attach with the channel, so it will definitely, protein also bring the charges. So when it brings charges, it will affect your channel properties. So let's say if you already have a proteins which are negatively charged, and now you bring a negative charge very close to the channel, so then your current which is passing through the channel will fluctuate. And now if there are some proteins, these are antibodies, so these can be used to capture actually different proteins from blood or other samples. So let's say if these are the proteins of interest which you want to sense, and now if some of those proteins will come and bind with your carbon nanotube, that will again change the local the local charge in this area, which will again affect the movement of the charge through the carbon nanotube. So again, you will see some change in the current. So it can be also used as a sensor. So this is a carbon nanotube based bio-FET sensor, and we'll talk about more later on, like what kind of antibodies can be used to capture different proteins from blood sample or serum sample or plasma. That will be discussed later in more detail. Okay, where we need these technologies, by the way. So there are quite a few areas, but we'll be covering mostly the areas which are related with the biology or human health. So in diagnostics, I have been talking a lot about sensing applications, but these particles can also be used as imaging contrast agents. Do you remember any technology which used particles as an imaging contrast? So these particles can go inside the human body and they can actually produce a contrast which helps us to see some of our structures inside our body with more clarity. Scanning, which kind of scanning use contrast particles? MRI, yes. So people use MRI for a lot of different applications nowadays, right? All this is very expensive. But in that case, we can use iron oxide particles, which is, iron oxide means it is magnetic particles. Magnetic resonance imaging, MRI. So those particles give a better contrast, because if you don't use particles, you can still see some structures inside, but not with that clarity. But when you use a contrast agent, they go and go to certain specific area, so you get a better image of the structure which you want to look at. And that's how MRI has been used in a lot of different applications for bone fractures, even for cancer diagnosis, and to see if there's an cyst inside a body or other structure. So it's a very, very useful technology. So yeah, these particles are also used for as a contrast agent, but that's also for diagnostic purpose. So those particles have been, so there are these nano-based technologies. They also have found a lot of applications in the therapeutics and drug pharmaceutical industry. In therapeutics, again, it's use imaging for the targeted drug delivery. Let's say these particles can be used as an imaging indicator to make sure that your particles are going where you want them to go. Let's say if you use an antibody, which is a drug. So some drugs are just based on the antibodies. So they go certain place, they bind with your cell or other structures. So to stop certain reaction, let's say if they're cancer cells, and if these cancer cells are growing a lot, and you want to stop the growth, there are some antibodies, they will go there and bind with the hormone receptors. So that will not allow cancer cells to grow more, for example. So in that way, you can stop the growth of the cancer cell to some extent, and that will be very helpful. But how we can make sure that the antibodies which we are putting inside our blood are going only to the specific organ, because in blood it goes everywhere. So for that purpose, sometimes people actually attach some tags with those antibodies, some dyes, so that they can go inside human body and you can see where they are going. So what are the important tags? So there are some inorganic dyes which are used. Most of them are fluorescent based, but it's very hard to image actually. When something is inside a body, on the surface it's easy to look at the images, like in the fluorescence even, but when it's inside, then it's hard, because then a lot of diffraction from the for the optical waveform which you use, and you cannot use a laser on a human body for imaging purpose, because you will be damaging a lot of other cells which you don't want to. So, and the signal which is coming from the fluorescence is not that strong, so it's very hard to see them. So people are looking at other kind of technologies which use wavelength of light, which like infrared based technologies, which can penetrate inside our body and will not harm other organs. But and the other thing is, if we are putting any particle inside our human body, it should be biocompatible. So if you are putting some dyes inside, and they might be toxic also. And that's why we do a lot of experiments. First people try with the animals, because there is actually right now no other reasonable option to test these dyes, and some animals actually die because of that, because at this testing phase before trying it on humans. So but some of the particles are very biocompatible, like gold particles, for example. They are normally considered biocompatible. So gold nanoparticles have been used for attaching with the antibodies and other drugs so that they can go inside. And gold particles give very good contrast at different wavelength of light. So you don't really need a fluorescent-based particles. So if you shine it with a certain wavelength of frequency, you will see a very nice contrast gold particles inside a body. Very strong signal. And these particles are also not toxic so far, what we have learned based on these studies. Why they are not toxic, by the way? Why people like gold, by the way? So why gold is very expensive as compared to other materials? Just color?
Doesn't oxidize easily.
Doesn't oxidize easily. It's a very, very good metal, and which is very, very stable. That's the one major reason. So if you make a jewelry, maybe after 1,000 years, still it will be in a similar shape. There might be maybe a very thin layer on the top of it, but it's very, very stable. And that's one property, actually. That's why it's also considered biocompatible, because once it's go inside the body, our body doesn't react, or these gold nanoparticles doesn't react with the solutions around it. So they stay stable. But yes, there are some questions still remaining which need to be answered. And those are where those particles go then. Because those particles might be staying inside our body for a long time. Whether it's good or bad, we don't know right now. So for long-term studies, people find that they are not toxic. But they might have some effect which show up after 5 years, 10 years, right? So you never know. And whether they are getting out, all of them are getting out of our body or not. So there are other options people are using. They are polymer-based nanoparticles. There's another example. Polymeric-based nanoparticles. They are polymers like PLGA. Some of them are also plastic-based, but they disintegrate after some time. So if you make a polymer and you make a particle, let's say PLGA, polylactic acid, this is a polymer, you can make a particles out of it, and you can attach like drug, but those particles doesn't give a contrast which you can get with the gold nanoparticles. But there are different other benefits which you get with the particles, that after some time, couple of days, they will dissolve in the blood, and then they can be actually removed from the body through kidney, through urine, so by themselves. That's a benefit, but again, it really depends for what purpose you are making those particles and why you are using it. If you use gold particles, people have used gold particles for a certain application, like let's say for local destruction of the cells. So in that case, they were actually attaching antibody that was going there into cancer cell site. But then if they are metallic particles, so they can actually use external energy source to transfer energy very locally to the gold particles, because their other media will not absorb energy. Certain wavelengths, I think 540 nanometer, they have a maximum absorbance, the gold nanoparticles. So they start getting heating up. So they will start heating up and they'll produce heat very locally. So if they are really at the place where there is a cancer cells, and if they are attached with the cancer cells, so they can kill the cancer cells with the heating effect. But the other cells will not be affected. So that's another way to actually destroy cells locally if you have particles which can be targeted to a certain, but that may not be possible with some other options. But nowadays people are also looking for another natural polymers. Let's say liposomes, for example. I told you that the cells have a surface layer in a previous lecture, right? And that surface layer is made up of phospholipids, right? Which is a hydrophilic and hydrophobic component. So those are, our body has many of cells, and all of that has a phospholipid. So it means the phospholipids are definitely biocompatible, right? And so if we make some particles or structures for drug delivery, for example, and if they are made up of a natural polymers which our body can take very easily, so that will be very, very good, and there should not be any biocompatibility issues in that case. And that's why people thought, okay, if we can have a cell membrane, so why not we can use a liposome, the phospholipids, like those molecules, and make a particle, small nanoscale particle with the technology out of that, and then we can use that for drug delivery. We can actually have a drug inside that particle and then make a particle. So there are different ways how you can make particle with the liposomes, but the point is that those particles can now be used to transfer drug to different parts of the body where you want them to go. You can also functionalize those particles. But the good thing about the particles is that they are not biocompatibility issues, they are not any toxicity issues, and those particles are very biocompatible. So yeah, there are different options in sensing and in also drug delivery, and also in other domains where you need nanoscale structures, features, and that's what we are trying to focus right now. Okay, let's talk about some solid-state fabrication. From now onwards, we'll go through some basics in silicon IC fabrication, the technologies which are used for making the transistors, and then later on, we will connect that, how we can use those technologies making devices and structures on nanoscale, which has applications in biology or medicine or in other biotechnology area. So some brief historical perspective. So these three people actually got a Nobel Prize in 1956 for their work, Baudin, Brittain, and Shockley. So they first invented the transistor in 1947. Then later on, Brittain and Baudin also demonstrated the oxidation process, the oxidation of actually silicon. So silicon is a very, very commonly used semiconductor material for a lot of devices, and more importantly, all our electronic components are made up of silicon. And silicon is you can get a lot of silicon with sand purification. So if you take a sand with some processes, you will be able to actually retrieve a silicon from the sand. So it's a low cost comparatively, and then it's a very useful material because it's a semiconductor, so you can make it a conductive or less conductive or more conductive based on how you dope it with external carriers. So by the way, but these are the first transistors were invented in 1947 with by these three people, and the transistor size was pretty big. You can see it, he's seeing it with a large microscope. So the size was this much big, one transistor. Now with that much big size. In that size, we can have actually billion transistor, or even more than that. Then later on, people start actually connecting transistors with one another to make some integrated circuits, or ICs. When we say IC or integrated circuit, that means you are connecting more than one transistor together to make that integrated circuit, which is actually now the modern day devices which we have, where we have billion devices or billion transistors all connected together to perform certain function. So the integrated circuit was invented in 1959 by Jack Kilby from Texas Instruments and Boyd Noyce from Fairchild, and they also got a Nobel Prize in 2000 for their work because they showed that these transistors can be integrated together to perform different functions. But at that time, it was at a pretty large scale. Okay, then some of the last century MEM nanotechnology timeline, for example, let me look at some good numbers. So the buckyball, which is a ball made up of a carbon, right? So nanoscale. Smalley was a scientist at the I think Rice University who discovered it. So they, along with another scientist, which I forgot the name, but buckyball were in 1985. So you can go through the list by yourself. So it's more about a historical perspective, when what was discovered and all that. So the carbon nanotube was discovered in 1991. So it's a very recent discovery. Before that, people just didn't have any information about the carbon nanotubes. Then 1996, Richard Smalley is the same guy who actually discovered buckyballs and develops a technique for producing carbon nanotubes for uniform diameter. Okay, there's another development. And then in 2000 or later, actually, people started looking more into using micro and nanoscale technologies for various applications in medicine and other areas. Okay, so this is a long list, so I actually want you to go through it by yourself. The slides are uploaded. Okay, so in the solid state fabrication, there is one very famous law. It's called the Moore's law. Do you know what's the definition of Moore's law? What this law says?
Double in power, half in size every 6 months.
Yes. So Moore's law says that the number of transistors in an integrated circuit doubles every approximately 2 years. Okay? So in 2 years, you can have actually twice number of transistors in the same surface area. So definitely you are increasing the power, right? So double every 2 years. Okay? And so in that way, if you see this trend as we are moving further, so these are number of transistors which we have on the surface of and you will see that 1970. So this is a trend which we have so far. So you are having more and more and more transistors in a small surface area. So far, people were able to follow this trend, but now it is start getting saturated, and there are reasons for it, because you can make things small, really small, and then really, really small, but to what level? It has to stop at a certain level, right? Because you can have a double number of transistors, you can make transistors small every 2 years. It's a law. It's kind of a prediction, not a law really, because he predicted that the technology will evolve such that every 2 years you will have double number of devices. Without any reasoning, he thinks that this is how the technology will evolve. Okay, so then it make it a law. They made it a law. So still, this law is being followed. Not really followed. How technology is evolving, this law is followed based on that automatically somehow. But now I told you that the companies, they are working on the technologies which are 7 to 5 nanometers for silicon fabrication and making those transistors and ICs. So yeah, you can increase the number of transistors, but then maybe after 5 you can work at even 1 nanometer technology by using carbon nanotube based structures, all that. But then what? After that, they are just atoms. How you can really deal with the atoms, right? So
how do they prevent quantum tunneling under 7 nanometers?
I don't know really. So very latest, there is no much information available on what exactly are the challenges which they are facing, but they are working on this technology. So when we talk about the 1 nanometer or down or smaller than that, then we cannot really go smaller than that, because you can have atoms and then what else? But now people are thinking about, there are some thoughts about quantum computing, where the idea is that if we can control the spin of the electrons, which are moving around the atom. So if we can control the spin, maybe each spin, because if we can have a control on spin, we can make many devices out of that. Let's say if we can control one spin, so if one electron is spinning one way and the other is spinning other way, and we can store some information in there, so then we can really store a lot of data even in one atom. But so that's another area people explore. I don't have much understanding of exactly what they are making with it, but yes, there are thoughts about it. that you can control the spin of the electrons, and you can build some transistors by controlling, and some storage devices by controlling that spin. As far as the storage of data is concerned, there is a very nice development about storing data in a DNA. You might have maybe seen some news recently, very recently. And there is also one company, it's a New York based, but I don't remember the name, but there was also in the news that they are working on this project. And why we really want to store data in DNA? Because if we have nowadays hard drives and all that, right, which can store a lot of data in small area and we can preserve that, right? But why the DNA-based data storage would be required?
Holds more data in a smaller space.
Yeah, DNA is small, but we are now reaching a level where we can actually control, we can have really small capacitors and transistors which are used to build those hard drive cells. So we already have actually I think a very good capability. You can have a small flash drive which is quite a few gigabytes, right? So which can store a lot of data. But the reason is historically, when people know that DNA doesn't actually disintegrate for thousands of years. So it's a very stable molecule on the other way, right? So when people discover new structures or dinosaur, for example, they find some new fossils. They tested that for carbon disintegration. But again, if there is some DNA there, they can test that. And that's how people also estimate, okay, how much humans are when humans were actually when the first human was on the earth. Like there are some estimations based on the scientific discovery, and one of the way to look at it is if we can find some sample which have some specific DNA with resemblance to the humans, right? So we can say something. So the DNA, if you preserve it properly and even in a harsh environment, it stores the information and it can store it for years. So in that way, why not? We can have data stored in DNA, and for the hard drives, they can, especially for the mechanical hard drive, they can damage very quickly. But in the case of DNA, yeah, they can also get damaged based on the different conditions. But yes, in certain conditions, it may survive better than the other methods which we currently use. And for DNA, again, we definitely have a capability to store a lot of data in a DNA, and then it's also very small scale. But again, now can we really store data? Why do you think we can store data in DNA? How we can store it, by the way? When I say store data in DNA, what does that mean?
Nucleotides.
What?
The nucleotides. Yeah.
With the nucleotides, yes. We have 4 base pairs like bits, right? So 1, 0. Normally we have assembly language, right, where everything is written in 1 and zeros. So you can transfer information. So then DNA, we can have 4 nucleotides, 4 different options. And you can store a lot of data in the DNA by controlling. So you have to define them first. Okay, when there is 3 A's, for example, what does that mean? That might be meaning alphabet A, for example. And if you write my name like Wasim, so W might be standing for A and T. So you have to define some language. And after that, you have to synthesize the DNA according to your language. So, but DNA synthesis is not very easy so far. But again, making things on nanometer scale was also not easy at one time. So I would never think about that we can have 1 billion transistors if I didn't know that it's happening. 1 billion times per 9 in small area. Okay, maybe if I was living in 1960s and 70s, I might think, okay, maybe it's not possible. So, but yeah, if we keep working on stuff, the things start getting possible after some time. So yeah, there is a development in that area also, controlling DNA sequences, and if we can synthesize it with a good repeatability and good efficiency, we would be able to store data in a DNA, and that can be preserved for a long period of time. Okay, before class finishes, we'll continue one more slides, but But I just want to announce one thing, that I'll be traveling for an NSF panel meeting. There's some duty which I have to go for some grant review on next week. So there will be no in-class session on Tuesday. But what I will do is I will record a lecture and I will upload it on a Canvas. So you should be able to actually watch the lecture, and they will also upload the slides. You should be able to watch the slides. So there will be no in-class on Tuesday, but there will be lecture which will be online on Canvas, so you can download it and you can watch the lecture. Okay, make sure you watch it on Tuesday, otherwise you will not watch it. Or maybe you will watch it before the exam, so it's better to maybe do it on the same day. We still have 2 minutes, so I will basically go through it and maybe a couple of more slides. So some of the background on the semiconductor materials. When I say, okay, silicon is a semiconductor material and it has been used for a lot of applications. Let me show you just periodic table, and then we'll continue from the next lecture with that, hopefully with that periodic table. So in periodic table, we have group 3, 4, and 5, which are more importantly used in a silicon IC fabrication. So group 3 has boron, aluminum, gallium, indium, titanium, and then carbon, silicon, germanium is a group 5, nitrogen, phosphorus, arsenic, antimony, and bismuth is a group 5, carbon is in group 4. So when we say semiconductors, it means most of these groups are actually conductive but semi. So they can be considered as conductors, and they can also be considered as insulators, partially conductive, partially insulator, especially the group 4, which has 4 electrons in the outer shell. Group 3 has 3 electrons in the outer shell. Group 5 has 5 electrons in the outer shell. So if carbon has 4 electrons, it means it needs 4 more electrons to fill its outer shell. Or it can actually lose 4 electrons also, right? So it's in the middle. So if there is something, let's say sodium, we'll stop there. I think we'll continue. Otherwise, yeah.